Okay, this is our podcast for Standard 3.1, which is all about the structure and function of DNA. So we will be looking at DNA molecule, looking at the general structure of DNA, as well as describing complementary base pairing. And we're going to look at how the location of DNA is in the nucleus, and that DNA is broken down in two sections called genes that are going to code for our different proteins, and those proteins determine our traits. Okay, so we're going to be using some new terms in here that you may not be familiar with, like adenine, guanine, thymine, cytosine. We'll be looking at the genetic code and looking at words like deoxyribose. So let's get started right away by talking about how DNA is actually a nucleic acid. So we talked in the beginning of the year about nucleic acids being organic compounds, and their job is to store and transmit genetic information. So anytime that you are looking at anything that is going to have to do with your genetics and how your genes are going to get passed on, you're actually looking at nucleic acids. DNA is the most common, nucleic acid in the sense that it's doing the storing of all of your information. DNA stands for deoxyribose nucleic acid. So there you can see your DNA, deoxyribonucleic acid. And another common nucleic acid is RNA, which is ribonucleic acid. And these two actually work together in order to store and transmit all of your genetic information. So when you look at these structures, DNA is this structure here. It's a much larger molecule. It's got two strands to it. And RNA is this one down here, which is going to be a single strand. So what we're going to be doing over the next few podcasts is we're going to take a look at how DNA and RNA are similar and different, and then how they end up working together in the process of transmitting your genes. So let's start by reviewing what a nucleotide is. Remember that nucleotides are the monomers of a nucleic acid. So remember, mono means one. So these are the basic subunits that a nucleic acid is going to be made of. All nucleotides contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. So all of those 5 different elements are going to be used when making a nucleotide. And all nucleotides have 3 main parts that are made up of these different elements. You have a 5-carbon sugar, which is different than glucose, which is a 6-carbon sugar. So this is a 5-carbon sugar. You have something called a phosphate group, which contains phosphorus and oxygen. And then you have your nitrogenous base. So the nitrogenous base. Okay, so let's take a look at what a typical nucleotide will look like. And this is just one version of a nucleotide. There's actually 5 different kinds of nucleotides. So if you were to take a look at a general structure, here you have your phosphate group. So you can see here's your phosphorus, and it has surrounded by oxygen. Here is your 5-carbon sugar, so 1, 2, 3, 4, 5. So that's your 5-carbon sugar. And there's a difference in the sugar between between DNA and RNA. In DNA, there is one less oxygen molecule on this part of the sugar than there is in RNA, and that's why it's called deoxy. So we'll take a closer look at that. And then coming up here is your nitrogenous base, and there's actually 5 different kinds of nitrogenous bases, but all of them contain an element of nitrogen within them. So nitrogen makes up a main part of them, so that's why they're called nitrogenous bases. So we'll take a look at what those different bases are. Okay, so in the DNA molecule, the sugar for DNA is called deoxyribose, where in RNA it is called ribose. And again, as I was just saying, the difference is right here in those 2 sugars. Everything about those sugars are the same, except that DNA, the deoxyribose, has less oxygen. So when you put D before something, it means without. So you can kind of think of the deoxyribose as being without oxygen. So it's got less oxygen than the ribose. That's the main difference, which is actually going to impact the way the molecule gets put together. They both have the same type of phosphate group. Again, you have a central phosphate surrounded by oxygens, and then this R is what it would go and attach to, where it would attach to the sugar in the molecule. Now, the nitrogenous bases are a little bit different. There's 2 groups of nitrogenous bases. The first group are called purines, and purines have a 2-ring structure to them. So there's two types of purines. They are called adenine and guanine. And then the other group are the pyrimidines. Pyrimidines are actually smaller molecules. They have only a one ring structure to them. And there are three types of pyrimidines. There's cytosine, thymine, which is only going to be found in DNA, and there is uracil, which is only going to be found in RNA. So we can take a look at what these bases look like over here. So adenine and guanine, those are your purines, and you can see that there are two rings to their structure. So their structures have these two parts to them. Whereas these two pyrimidines that are being shown, cytosine and thymine, they have only the one ring structure. So they are smaller molecules. This is looking at the bases that are found in DNA. And we know that because this is telling us it's deoxyribose. But uracil also has a similar structure as cytosine and thymine, where it's a one-ring structure like that. Okay, so here's all of the different nucleotides put together. So you can, again, here's the uracil that we were just talking about. The sugar on this one is ribose. But again, it's that one-ring structure like the thymine and cytosine, whereas adenine and guanine both have a two-ring structure to them. All right, so we're going to talk a little more specifically about DNA. DNA's job is to store and transmit genetic information, and this happens in all living things. Every single living thing, one of the fundamental characteristics of life is that they contain DNA. Long pieces of DNA are called genes, and genes are going to determine the proteins that a cell can make. And those proteins that they make are your actual traits that you see. So your hair color, skin color, how tall you are, all of those types of things. Okay? And so remember, genes are what actually make up our chromosomes. So you can kind of look at it in this picture here where you have got your chromosomes inside of the nucleus. Remember, in a human you would have 46 chromosomes. Now here's looking at one of those chromosomes. One chromosome right here is made of DNA. If you unravel that DNA and you look at just one little piece of the DNA, that is a gene. So one little piece of this entire chromosome is a gene that is going to determine what kind of proteins you make and thereby what you would look like. And that's what we had just studied previously when we looked at all of Mendel's traits and all of those kinds of things. So DNA structure. DNA has a backbone of alternating deoxyribose and phosphate groups. So here you have a phosphate sugar, phosphate sugar, phosphate sugar. On both sides of this molecule, we call that the backbone. And those sugars and phosphates will repeat over and over again, and then connected to them in the middle are the nitrogenous bases. The nitrogenous bases are going to connect to each other by something called complementary base pairing. This is referred to as Chargaff's rule. What we know is that purines always complement pyrimidines. That means that they're going to bind to each other. So adenine is going to bond only with thymine, and guanine is going to bind only to cytosine in a DNA molecule. Okay, so A always binds with T, and G always binds with C. You actually never find A and C together, and you never find G and T together. And the way that Chargaff figured this out is he actually looked at a DNA molecule and figured out that there is the same number of A's and T's in the molecule and the same numbers of G's and C's in the molecule. So he figured out that A's and T's were always together and G's and C's were always together. This is going to be really important when we talk about replication, because it's going to make sure that there's not very many mistakes that are going to happen during the process of making copies of our DNA. Okay, so as those DNA nucleotides come together, we end up forming what is called the double helix. DNA is composed of two strands. So here's one strand and here's the second strand. And there are hydrogen bonds that are between the adenine, thymine, guanine, and cytosine that are going to hold those two strands together. So between the adenine and thymine, there are two hydrogen bonds that hold them together. And then between the guanine and cytosine, there are three bonds that are holding them together. And it's those chemical bonds that keep that molecule stable inside of your cells. And it's only when the molecule is going to replicate or to make some RNA that those bonds can be broken, and then we can open up the molecule and we can use it. But the fact that they're hydrogen bonds that are holding them together, hydrogen bonds are not the strongest of the bonds, and they allow for some flexibility. So once the molecule has been formed with these bases, it then has the ability to twist into this double helix. That twisting motion is actually going to make the molecule more compact, so you can fit all of it in the nucleus of the cell. So that's where these two strands are going to then twist around each other, and you get what was referred to as the double helix. Some people call it a winding staircase. You can think about it like almost like a spring in a sense, but it winds around each other, because if you unwind this, it would be a very large, very long molecule. So by twisting it, you make it much smaller, so it will fit, makes it more compact. Okay, so here's another picture of DNA just showing you the bonds that occur between them and how they are held together. So we have our deoxyribose and phosphate. They make up the backbone. They are alternating all the way down the molecule. Connected to the sugar, the deoxyribose, is the bases. And then your thymine and adenine are always going to bond to each other with 2 hydrogen bonds. Guanine and cytosine are going to bond to each other with 3 hydrogen bonds. The order of these bases that are here, that is what was going to determine what kind of protein we can make. So what we call that is the genetic code. So it is the sequence of the bases in the DNA. We call that the DNA sequence that's going to determine the actual protein sequence and thereby the protein structure. So it is the sequence of those A, T's, G's, and C's, whatever order they're in, is going to determine what kind of protein we can make. So when we talk about these nitrogenous bases, we actually are going to end up arranging them into triplets called codons. So a codon is a series of 3 bases together, and then each codon is going to correspond to one of our 20 different amino acids. So there's 20 different kinds of amino acids that make up proteins, and depending on the order that those amino acids are in, that's going to determine what the protein can do, how it functions. In order for the cell to know what proteins or what amino acids to bring in and in what order, the DNA is holding on to that code in these things called codons. So this is what we're going to be investigating over the next few podcasts. Now we also have special codons that are going to mark the beginning and end of genes, so we know where one gene starts and another one ends, and these are called stop and start codons. So that we always know where we have to begin making a protein, we have a start codon, and when we need to stop making that protein, we have a stop codon. And that way we can always finish the process of making the protein completely. So in this next slide, this graphic is going to show us what this is going to look like. A common start codon that is always exists is TAC. TAC also codes for methionine, which is one of our amino acids. So you can kind of think of it as every protein starts with amino acid methionine, although a lot of processing happens with the proteins once they're made, so that's not necessarily true, but TAC is always going to be the start codon, so it tells us where to start making that protein. So we can look at the genetic code in this way. Here we have a DNA sequence, and we read that DNA sequence in 3s, so kind of like reading a sentence. So CCC, TGT, GGC, so that's how we read it. And then each of these triplets are going to correspond to one of these different amino acids until we get to one that says stop. So here's another graphic of that, kind of telling you what we're looking at. So here's our DNA. Okay, we have a piece of that DNA right here, and this is the code. And we read it in 3. ATC is 3 bases that are going to code for a specific amino acid. CTA, code for a different one. GGC, code for a different one. And there's always something that's going to tell us to start reading it here, and it's going to tell us to stop reading it somewhere else. So again, when we get into the next podcast, we will start to take a look at how that process can happen.